Hello, my name is Mr. Communell, and today I'm going to show you how to do the BioRad pGlo lab. This is going to include teacher setup, students running the experiment, and then lastly, the results. This is a little bit of a challenging lab. At some points, you just got to make sure you maintain a sterile environment when you're working, but at other points, you have to make sure everything is kept at a certain temperature, mixed properly, and then parts of the lab, make sure you are timing some of the processes. So this should help you out in making sure that you don't make any crucial errors that would affect your results at the end of it, and hopefully your students have a successful transformation. When you're doing the BioRad pGlo lab, you want to start off by growing the bacteria. The directions, the procedure are going to tell you to plate it first, but I like to get my bacteria growing fast so I can generate more colonies whenever the students want to plate their bacteria. So I already did this, but I will show you how to do it. I have my E. coli that you get in the packaging. Hopefully you refrigerated this and it's stored at about 4 degrees Celsius. I also have my LB nutrient broth here. And what we're going to do is we are going to transfer 250 microliters of our LB nutrient broth into our E. coli, okay? So again, I already did this, but you want to make sure you're using a brand new sterile pipette. I'm not going to take one out of here because these are all sterile, but you do want to go to fill the nutrient broth to the first line on your pipette. And that first line represents 250 microliters. So you will take this, the nutrient broth, and place it into here. You'll swirl it around a little bit, all right? And then the last thing I want to do is, I like to do this, you do not have to, but I like to put it in a beaker, a small beaker or something like that while it's in the incubator. But once you have it like this, and that's the exact reason why, because if it gets hit or anything like that, it's not a big deal because I have it in the beaker and it's not going to completely topple over. So once you have that, I'm going to go to my incubator here. I have it set for 37 degrees Celsius. And I'm going to place that in. And again, that's another reason why I always like to have it in a beaker or something like that in case it falls over. Now, one more thing I want to mention with this, and you can see it coming up to temperature. I'm going to have it at 37 degrees. If you do not have an incubator, what you're going to want to do is let this grow for probably 2 to 3 days before the students do the experiment. And this is just going to generate more colonies that the students will get on their LB plates. One more thing I want to mention, just how I do it, is I will start this on a Friday. So I'm growing the bacteria. Today is Friday, and I'm going to let it grow all weekend, even in the incubator, just so students get more colonies. Okay, now we are going to begin plating. I have the BioRad pGlo instructions here. I cleaned off this table so it is completely sterile before putting my plates down, and then I labeled my plates. The instructions say to do 16 plates that are just LB broth. It says to do 16 plates that are LB broth and ampicillin. And then it says to do 16 that are LB broth, ampicillin, and arabinose. Now, I'm not doing that many this year because I don't have a class that big, so I may as well conserve resources. Again, we want to make sure everything is sterile. I have the micropipettes that BioRad gave us, and they are sterile as well. They're still sealed. Before I start plating, and I already have my broth made, I'm going to begin mixing the ampicillin and the arabinose. And why I'm going to start doing these now is because the arabinose takes a little bit of time to dissolve. So we're going to take this, and they give you transformation solution. So we're going to put 3 milliliters of the transformation solution into each one of those. So I'll start off with the arabinose because it takes a little bit longer to do. So you just pull this top off, okay? And then we're going to get our transformation solution, which is calcium chloride. Now, with our micropipette, and you'll use this throughout the lab, you have 250 microliters, you have 500, 750, and then up here is going to be 1 milliliter. So you're going to want to put 3 of those into your arabinose and 3 of those into your ampicillin, measuring each out at a milliliter. Okay, so I'm going to unpop my arabinose here, go up to 1 milliliter, 1, 2, and 3. And I'm going to close these up, both of them. And I'm going to throw this pipette away. And then your next thing is going to take about 10 minutes. You just want to swirl, 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 swirl. Okay? So I'll swirl this, and then we'll do the same thing with the ampicillin. So I just finished mixing my arabinose. It did take a little bit over 10 minutes. The solid was very resistant to wanting to dissolve, but just be persistent on it and it will dissolve. So now I'm going to do the ampicillin next. So once again, take the top off. Okay. And we're going to get a brand new pipette for this. And once again, we are going to put 3 milliliters of our calcium chloride in here. So 1, 2, and 3. And we will throw that pipette away once again. You do not want to reuse these things because it will get contaminated. And now we're just going to mix this. This one will not take as long, but it still might take a few minutes to get all of these solids to dissolve. Okay, we are done dissolving the arabinose and the ampicillin. So now we're going to move and start to plate our LB broth. Now, BioRad does give you a packet of LB agar powder, and if you wanted to use this, all you're going to do is dissolve this in 500 milliliters of distilled water, and you should use about 1,000 milliliter Erlenmeyer flask to do that. Why I would use an Erlenmeyer is because the top of the Erlenmeyer flask is going to be very small compared to a beaker, so that just minimizes your risk of contamination. But I had some leftover LB broth, so I figured I'd use this. Plus, it's a little bit less than what the packet makes. So since I'm not plating as many plates this year, I can just try to conserve resources. So to plate, we're going to do all the ones that are just labeled LB right now. So you're gonna label, if you're following the BioRad instructions, you're gonna label 16 with LB, and we are going to plate each one with the LB agar. Now, when you're plating, you're going to quickly pull this off, and you're going to fill it about a third of the way or halfway up with your broth, okay? So you wanna kind of be ready. You're going to pull this off, and cap. Now, one thing that's important for later on, if you're noticing, I plated 2 of them, and you can see that there is a fog coming on the top of them, and that's going to be condensation. So once these are completely solidified, what we're going to do is we're going to flip them upside down, and what that's going to make sure is all that condensation going to the top doesn't fall off and go on our agar plate. So I'm going to continue this process to plate all of the LB broths, and then we will next move on to the LB broth mixed with the ampicillin. So I just finished plating all of our LB plates, and now I'm going to move on to the LB ampicillin plates. So we're gonna plate these next. The pGlo instructions say to plate 16 of them, but you can change that depending on how many students you have. So what we're gonna do first is we are gonna add our ampicillin that we mixed up to our remaining LB broth. The important thing here is that the LB broth cannot be above 50 degrees Celsius. If it's above 50 degrees Celsius, our ampicillin is not going to work. So we're going to take this off and we are going to pour this in. And the other thing to remember is that the LB broth is also going to solidify around 27 degrees Celsius. And since it's going to solidify at that, you have a very narrow temperature range. So we're going to mix this up a little bit. Get our ampicillin all mixed. And then once it's mixed, you are going to plate again. So we're going to have your broth about 1/3 of the way up, and then cap it, okay? And then you'll do that with all of the remaining plates. So I'm pretty much done plating my LB with my ampicillin on my last one here. And after that, we are done with those plates. So my last one I'm gonna plate is gonna be my LB, ampicillin, and my arabinose. So we're gonna label the pGlo lab instructions say to label 8 in that manner. But before we can pour, we gotta add now our arabinose to our LB broth. So I'm gonna take this, we're gonna pour that into our LB broth, and we're gonna stir it around a little bit. And again, same thing with the ampicillin. You wanna make sure your agar is not above 50 degrees or the arabinose will not work. So let's plate our last little bit here. And we'll continue plating until all those are done. Now, after everything is plated, so you'll have all your plates, I do this on a Friday. I do my prep on a Friday. And then I have the students come in and they can start working on Monday. That way we should finish the lab by Wednesday. And why I do this is because I want the bacteria to have time to grow over the weekend. Now, along with that, it also gives your plates a longer time to get contaminated by bacteria, which you also don't want. So what I would do is if you are prepping this on a Friday, you can either A, come in early on Monday and prep your plates, or B, prep them with the bacteria on Friday and then just put them in the refrigerator. They can be at about like 4 degrees Celsius and they should be free of bacteria whenever you come in on Monday. And that would just save you some time with coming in early. Okay, so I let all of these solidify for about an hour and a half. And then after the hour and a half, I flip them all upside down so that the gel is on the top. Because if you look at the bottom, all that condensation from the agar cooling is still there. And I do not want that to get onto my gel. Now, if you wipe it with a paper towel or something, you are still making it susceptible to bacteria and it could possibly be contaminated. So we're just gonna leave that condensation there because it's not gonna bother us. Now we're gonna make our packs for our students, but one thing to consider before we make our packs is how do you want the students to go about doing this lab? You can either grow a bacteria plate for each student, or each group, or you can have the students do it on their own, and that's the option I like to do. So you already are incubating your E. coli at 37 degrees Celsius in the incubator, and I'm gonna let that go over the weekend so I get a lot of bacteria multiplying in that container. Come Monday, I'm going to have the students plate that bacteria and let that grow on Monday, and then they can grab it on Tuesday. So what I'm gonna do now is I'm gonna make packs for the students. Now, if you are having, if you're going to grow the bacteria, here's what you would need. You would need in each group 1 LB plate. You would need 2 LB ampicillin plates. And then you would need 1 LB ampicillin and arabinose plate, okay? Now, I'm gonna have all these plates, they're all gonna be upside down, and what we can do is get a piece of tape and tape them together. However, I'm not going to go this route. I'm gonna have my students grow their own bacteria and use their own colonies for the whole experiment. Given that, I'm gonna get 1 more LB plate, and I'm gonna put that in their pack, okay? So they're only gonna be using, on Monday, the LB plate, that single solitary one. And then come Tuesday, they're gonna use their product of this and plate that onto here, but that will be on Tuesday. So right now, they're only gonna need the LB plate, but I'm gonna put these in the refrigerator so that no bacteria grows on them over the weekend. So I'm gonna get my 5 plates here, and I'm gonna put a piece of tape around it. And I have my student pack already, and I'll keep these refrigerated over the weekend. Then on Monday, they're gonna pull out their 1 solitary LB plate and allow that bacteria to grow on that medium and grow for a day. So we're now ready for our next step. Now your E. coli has been growing in an incubator for 24 hours at least at 37 degrees Celsius. If you don't have an incubator, I would do at least 2 days, just sitting at room temperature. And you have to make a decision now. Do you want your students to make their own original bacterial colony plates, or do you want to do it for them? I have my students do their own, and then they use their own colonies for it. This way too, if one group doesn't get colonies and all the other groups did, they could just borrow some colonies from another group. So what you're gonna need for this is an LB plate, okay? Before we plate it, let me go over the proper streaking techniques for this plate. So what you're gonna do is you're going to get a loop, and in your BioRad kit, you're gonna have a bunch of loops in there. And what we're gonna do is we're gonna use one of those loops, we're gonna dip it into our E. coli, and then we're gonna begin the streaking process. So what you're gonna do is after you have it into your loop, pretend this is your LB plate, you're gonna start here with your loop, and we're gonna divide this into 4 quadrants. So our first quadrant's going to be just taking that loop, putting it gently on the agar, and going back and forth, okay, in one area. After that, using the same loop, you're going to go through this area to make quadrant number 2. So we're gonna go up, and I like to go back through, but not through the whole thing, just to get a few more colonies from this area. And we're gonna go up and back, just like this, okay? To do the next quadrant, we're gonna do the same thing we did with quadrant number 2. We're gonna go through, back a little bit, and carry on with that loop, okay? And then to do quadrant 4, again, same thing, up, back through, okay? Something like that. Now, your quadrant number 4, which is going to be right here, this is probably where the students are gonna get the best, most isolated colonies for the next few steps. So again, your thing right now is just to determine, do you want the students to do it, or are you gonna do it for them? So after you are ready, and if the students are going to do it, what you're gonna do is you're gonna get your LB plate, okay? And you're going to get a new loop. I have some old ones. You'll be using these ones, okay? Now, the important part is to not touch the actual loop. It is sterile at this point, okay? We're gonna open our E. coli. We're gonna take our loop out. And what you're going to do is you want to slightly just tip this sideways a little bit, okay? And then you wanna get a real good amount of your E. coli on there. You can shake your tube a little bit just to make sure all those cultures are dispersed evenly, okay? And then get a good bit on that loop. Now, you only need the loop. We don't have to be, putting a certain amount of microliters onto our plate. After that, we got our plate here. You're going to open your plate, take your loop out, and then we are going to begin streaking. Okay. So again, just the same format we did, very gently. Okay. And then we are going to carry this out into our next quadrant here. Go back through a little bit. Okay. And then the next area, come through. Oops. I punctured the gel a little bit, not a big deal there. Okay, I'm just gonna avoid that little area. And then we're gonna go through the last area right here. Okay, so right in this quadrant 4 right here is probably where we're gonna get our best cultures at, as I said. And then your next step, close this, let that dry on there for about 2 minutes. And then as it is, or after it's done drying, what you'll do is you will flip this over and you're gonna put it in the incubator, again, 37 degrees Celsius for a day. The one disadvantage of having the students do this is all they do during that first day. They're gonna plate it, they're gonna put it in an incubator for 24 hours. So I would have something else planned for class that day because it's only gonna take like 15 minutes for them to do. If the teacher does it, you don't have to worry about this step and you kind of save a little bit of time for your students. So everything is set up for the lab and it is now the next week. The students on Monday, they came in and they plated their bacterial plates. Now you can do this on your own if you would like and have the bacteria already grown on an LB plate, but I have the students do it. Here's one group's LB plate. They took the E. coli bacteria, they grew it for a day, we put it in an incubator at 37 degrees Celsius, and we let it for about 24 hours, and these were the results we obtained. So now they're gonna start on the actual procedure part of the lab. I already have ice ready for them. You will need ice today. You're also going to need some hot plates, and I have water in the hot plates, and it's set at 50 degrees Celsius. I'll show you more of that later. But what we're gonna do first is we are going to rehydrate our pGlo. I do this the day of the students actually doing the transformation. Reason being is you gotta put this in the refrigerator after you hydrate it. But if you do it that morning, you don't have to worry about putting a bunch of stuff in the refrigerator. So what we're gonna do is we're gonna take our pGlo plasmid. It does not look like anything is in there. I promise you something is in there though. It's just very, very small. We're gonna need our sterile pipettes, and we're gonna need the transformation solution, which is calcium chloride. So what we're gonna do is we are going to get 250 microliters of our calcium chloride. And recall that is the first line on our pipette. And we're going to put that into our pGlo plasmid. Now for this, you're gonna wanna shake it up. You don't know if this has been tossed and turned, so that plasmid could be on the top of your vial here, so shake it up real good and do it every which way. And now this will be ready for the students to use. You could just let that sit out if the students are going to be using it fairly soon afterwards, but if it's going to be a good bit later, you're going to want to refrigerate that. Our bacteria has been growing for 24 hours now. I just pulled it out of the incubator, and this is what it looks like. You can see the different quadrants there, and up at the top in quadrant number 4 there, you can see I got some pretty good isolated colonies, and that is where we want to use those colonies to do the transformation. So before we are going to have the students do the lab this day, a few things we want to do. First thing is we want to have a hot water bath, and this should be at 42 degrees Celsius. Again, that's 42 degrees Celsius. The other thing we're going to need for this day is going to be some ice. Now, let me show you how I do the ice here. Instead of going to the store and buying the little, I guess you want to call them ice crystals, almost like small little bits of circular ice, I just get regular ice. And then what I'm going to do is I just hit it off my lab desk to break it up into smaller pieces, and that increases the surface area of my ice. So I'm not going to do that on camera because it's pretty loud, but once you have that all crushed up, you're going to put it into some kind of small container like this. So now I have my ice all crushed up, and I'm going to put that into my little glass container here. And by crushing it up, what this does is it just increases the surface area that our little microvalves are going to have to be in contact with the ice. If you use the big pieces, they might not be completely surrounded by ice. You do not need a whole lot of ice. This was like a quart-sized bag, and I still have at least half of it left over. So now you can kind of just let that sit for a few minutes while we prep the next pieces. You're going to get these little microvalves here. I labeled one negative pGlo, and the other one is going to be labeled positive pGlo. And you're just going to set them in this little styrofoam container that the BioRad kit gives you. The next step is you are going to need the calcium chloride. That's the transformation solution. So you're going to need that next. And what we're going to do is we're going to put 250 microliters into each of our microvalves. So go ahead, get a sterile pipette. And again, just as we did in the previous step, it's 250 microliters. So we're going to put 250 in this one. And then 250 microliters in the other pGlo one. After that, dispose of your pipette. So now we're going to get our LB plate that we plated the bacteria on, and we are going to get 2 loops for this. So you're going to need 2 loops. And what you want to find is you want to find a very nice isolated colony of bacteria. So I'm probably going to use the ones right around here. Okay, so you're going to just take that and get it onto your loop. Okay, so you won't be able to see it on the loop, but it's gone from there, so it is on my loop. And you are going to put that into one of your pGlo ones, and you're going to stir it in there. Just go back and forth. You can revolve and rotate it around. Okay. And then this, we're going to throw this away now. Get a fresh loop. We're going to get another colony and put it into the next pGlo one. So I have a new loop here. I'm going to use this colony right here. Okay. Put that in your other pGlo, and you're going to revolve, rotate, mix it around in that transformation solution. Okay. Okay, your next step after this, and cap them just to make sure that they do not get any contaminants in them, okay? Now the next step, we're going to get our pGlo plasmid. Now this is important because we have a positive pGlo and we have a negative pGlo. So what we want to do is put 10 microliters of our pGlo plasmid into the positive pGlo only microvial. Here is my micropipette. I have it set for 10 microliters and I have my pGlo plasmid, okay? We already put the solution in here and we mixed it around, but give it one more mix, okay? Just to make sure that plasmid is all evenly distributed, okay? And again, we're only putting this one into the positive pGlo. So we're gonna uncap it here. And we are going to put that in, okay? So after you have that in, recap it. We're gonna mix that around a little bit. And then what we're going to do is put that on ice for about 10 minutes. So you could take them right out of your little styrofoam container, and we're gonna put that in. Make sure to pack the ice real good around both of these, okay? Again, this is why we crushed it up. So pack the ice real good, and we're gonna let that sit there for 10 minutes. So after they have been on for 10 minutes, what you're gonna do is get your little styrofoam holder again. We're going to take these out, put them in. Now, I'm gonna pick it up and push these all the way down as far as I can. I'm gonna do the same thing with the negative pGlo one, okay? And what we're gonna do is we are going to put this in a hot water bath for 50 seconds. So that's your next step, 50 seconds in the hot water bath. Once 50 seconds has gone by, remove it from your hot water bath. We're going to return it to the ice. And we're gonna put it on ice for another 2 minutes. Again, make sure to pack the ice around it real well, just so that ice is in contact with your solution, and let it sit there for another 2 minutes. After we've let it sit for 2 minutes, we're going to take these out, try to shake off the excess condensation from the ice, and we're gonna return them to our styrofoam. So now what we want to do is we want to let it sit for 10 minutes, but before we start that, we are going to get 2 sterile pipettes and we are going to get our LB nutrient broth. And to each one of these, we are going to add 250 microliters of LB. So open your first one up, add your 250 to it. And again, make sure to get a new pipette each time. Swirl that around for a second or two. And what you're going to do now is just let them sit for about 10 minutes. Now, while they are sitting, here's what you want to do. You're going to give your students 4 plates. You're going to give them an LB plate. You're going to give them an LB with ampicillin plate. I just labeled AMP, but there is LB in it as well. You're going to give them 2 of those, sorry, actually, 2 AMP plates. And then lastly, you're going to give them an LB, ampicillin, and a arabinose plate, okay? So when we have these plates, you want to label them So on the LB one, we are going to label it negative pGlo, just like that. Okay, so we want to put on the LB plate negative pGlo. On one of your, I relabeled it there for you, LB ampicillin plates, we are going to also write negative pGlo. Okay, so negative pGlo on that one. On the other LB ampicillin plate, we are going to write positive pGlo. Okay, so positive pGlo. And then our last one, the LB with the ampicillin, the arabinose, we are going to label that one positive pGlo. Okay, so in the end, and here's how I like to set these up just to make it easier on my students, is we're going to have our two negative pGlos, our LB and our LB ampicillin ones. I like to put those up top because you know both of those are going to go with our negative pGlo plasmid, okay? And then the bottom ones I like to put together for our positive pGlo because that one, we're going to put this one microvalve onto those two, okay? So while you're waiting the 10 minutes, you can have your students label these plates. Okay, so we are now on our last step for the students. Now what you're gonna do for this step is you are going to put your transformed plasmids onto your plates. In doing this, what you're gonna need is you're going to need 4 sterile loops, and I'm gonna use a micro pipette. Now I have this one set for 20 because it only goes to 20, and I do not have a bigger one, but we are going to put 100 microliters of our solution on each one. So the negative pGlos, we're gonna put 100 microliters of the negative pGlo one on there, and then our positive pGlo, we're gonna put 100 microliters on each one and then streak them. So what I like to do is I like to do them individually. That way the solution isn't going to get absorbed in the agar in one place. So I'm going to put my 100 microliters on my LB negative pGlo first. I'm gonna streak it, and then I'm going to carry on to the next ones. So I'm gonna flip this first, and the streaking is gonna be a little different with this one. We're going to just do a full spread on this, and what you're gonna do is you're going to get your negative pGlo here, and I'm going to, because I'm doing 20 each time, I'm going to do 5 of these, not directly in the middle, slightly to the side. Okay. Recap your negative pGlo, put that back, go ahead and get a sterile loop, and what you're gonna do is you are just going to spread it throughout the whole thing, okay? Turn it 90 degrees, do another spread, okay? If you wanna do 3, you can do 3, okay? Just go very gently on the agar, all right? After that, cap it. We're gonna let that sit for about 3 minutes before we flip it over. And then while we're doing that, get another sterile loop, a new micro pipette tip, and we will now do the LB ampicillin negative pGlo one, and we'll repeat that process, okay? Get a new sterile loop, and very gently, sorry for the bell, but very gently spread that. Yeah. So we'll do the same process, but for the positive pGlo ones, you're going to want to use these. The positive pGlo microvalve. So again, get a new micro pipette tip. You're going to flip this one over. Now we're using the positive pGlo. 1, 2, 3, 4, and 5. Okay. And again, I'm doing 5 because I'm just using 20 microliters each time I do it. All right. And then you can repeat the process with the last one. I won't add more time to this. But after that, let them sit for 3 minutes, and then we'll flip all of them over so that the gel is up. So I now plated all 4 of them. I let it sit for about 3 minutes just to let that solution on there dry. And now we are going to flip them upside down. And you can stack them on top of one another. And now we're going to incubate them for 24 hours at 37 degrees Celsius once again. So this will be, if the students are making their own bacterial cultures in the beginning, this will be day 2. So all of this part is day 2. Day 2 takes a little bit of time for the students to plate and do everything, just because they're a little slower with the streaking, because they usually don't streak agar plates. So give them a little extra time on this day. I know I went through it fairly quickly, but it'll take them a little bit longer. So again, 37 degrees Celsius, let that sit for 24 hours, and then we'll come back and see what our results were. Okay, so these are the results the day after. I have these uncapped just so you can see it better, but my regular LB negative pGlo, we definitely got growth in there, okay? My LB negative pGlo with ampicillin added, there's no growth in that one. My LB ampicillin with the positive pGlo, you could see we got growth. So the negative pGlo, we didn't, but the positive pGlo, we did. It's the same exact plate for these two, but one had growth, one did not. So this tells us that adding that pGlo plasmid is going to give us antibiotic resistance, because the ampicillin antibiotics cause the bacteria not to grow in that one. And in our last one, these plates look fairly similar, a few more colonies in this one with the arabinose, but we have, again, our LB ampicillin one here with the positive pGlo, and this is our LB ampicillin arabinose with the positive pGlo. And you can see we got a lot of growth in that one too. So let's see now that, yeah, definitely the positive pGlo gave us the antibiotic resistance, but let's see if also adding that positive pGlo with the arabinose is going to allow us to give that gene expression of the glowing factor. Okay, so brought a black plate with us here. Our first plate, we have our regular LB, and the bacteria did grow, but it's not glowing. Our second one, well, we didn't have any bacterial growth at all because of the ampicillin, so no growth, obviously no glowing. Our third one here, we have our positive pGlo, and we also have the ampicillin present, which we did get bacterial growth, but there's no arabinose, so we did not get any kind of glowing there. But the fourth one here, there you go, we have the arabinose present. We have the growth of the bacteria. It was antibiotic resistant because we had the ampicillin there. And the arabinose allowed us to express that pGlo gene, and that's why we have the glowing. So without the arabinose, we would not have any kind of gene expression. So hopefully this little tutorial helped you out, and hopefully your students are successful as well with getting that positive pGlo transformation.